Okay, so we're looking at the two steps in protein synthesis: transcription and translation. So in transcription, let me get my pen here. In transcription, the complementary base, the code that's in these complementary base pairings holds code for making the proteins. So the base pairs, remember, in our DNA are adenine, paired up with thymine, and guanine paired up with cytosine. Okay, so let's look at this in a little bit more detail. So what's happening here is we're transcribing the information that's in the complementary base pairing and transcribing that into an RNA molecule. So the main player here is this enzyme, RNA polymerase. Here is our DNA strand, and this strand right here is going to be our template strand. So the RNA molecule is going to be made following the pattern of the template strand. This area of DNA, so within this area right here, there is a sequence of nucleotides. There's a sequence of nucleotides in what's called a promoter. And it's this promoter that signals where RNA polymerase should start transcribing a specific gene. Okay, and let's look at this in even a little bit more detail. So here is our RNA polymerase, and our RNA polymerase is unzipping the DNA molecule. So this is a little bit different than when we were looking at DNA replication. In DNA replication, the whole strand, DNA strand, is unzipped and replicated. In protein synthesis and in transcription, only the part of the strand that holds the gene, the sequence of nucleotides that are specific for this gene for making a specific protein. So only a certain part of it is unzipped. And in this picture right here, you can see that this area right here was the promoter sequence. So our RNA polymerase started way back here and is going in that direction and unzipping the DNA double helix as it's going, and creating an RNA chain using this template of nucleotides. So there, in an RNA molecule, are complementary base pairs were, so what do we say? So we said guanine and cytosine, and thymine and adenine. In an RNA molecule, there is no thymine. Thymine is replaced with the nucleotide uracil. So instead of thymine, let's see, instead of thymine being paired with adenine, uracil is paired with adenine. Okay, so it's adenine and uracil, and the other two are still the same as with the DNA. So guanine and cytosine. So what we've got right here is our RNA polymerase matching up a uracil with an adenine, like we've got right there, an adenine to a thymine, a guanine to a cytosine to a guanine, uracil to an adenine, cytosine to a guanine, to make this RNA strand. Okay, so this attraction right here with RNA nucleotides to DNA nucleotides is actually very weak, much weaker than the attraction for DNA nucleotides. So our DNA molecule, our double helix, is a very stable molecule, which means that it doesn't react very easily with any other molecules. But DNA, this strand right here, and the RNA, this temporary molecule right here, is not very stable at all. And that's why, as the RNA polymerase passes through and forms these, helps to form these bonds, as it passes, the attraction breaks and the RNA tends to, what's the term? There's a really funny term. What was it? Dangles. That was it. That the RNA molecule dangles off the DNA strand. And because the DNA nucleic acids, these nucleic acid chains are very stable together, it zips back up behind the RNA polymerase. Okay, so let's see. Let me look at my notes to make sure. Oh, the other thing that's different is, okay, so in an RNA molecule, there's a uracil instead of a thymine. The other difference is the sugar in an RNA molecule, instead of a deoxyribose sugar, there's a ribose sugar. So these are called ribonucleotides. Okay. And again, just like in DNA, our ribonucleotides are added to the 3' end. So you can see that right here. We've got our 5' end of our RNA molecule here. And then at this tip over here, this is going to be our 3' end. So that we know, boy, does that look like a 3? There we go. 3' end. We know there's a hydroxyl group hanging off right there for this new nucleotide to be added on. Okay, I think that's pretty good on transcription. Let's take a look. Okay, so now we have, oh, let me go back. So then the RNA polymerase comes to a sequence of nucleotides that signals the RNA polymerase to stop. And as soon as it stops, this RNA chain will then fall off the RNA polymerase and off the DNA. And before that RNA can go outside of the nucleus, it needs to be modified. So in this DNA, or excuse me, RNA now, so here we have our RNA molecule that was just transcribed. But in this chain, there are sequences called exons and sequences called introns. The introns are actually sliced, and then the exons are spliced together to form the final RNA molecule that's then going to go outside into the cytoplasm. So these exons are coding. So this is, that's weird. My pen keeps disappearing. Give that another try. Okay, so so this is coding, protein coding. These introns, so protein coding, these introns are non-coding. Sequences of RNA. And this is one of the examples of the non-coding sequences of DNA and RNA. Okay, oh, and also a cap is added, and then a poly-A tail is added. Okay, and now it's ready to go outside of the nucleus and into the cytoplasm, where we can talk more about translation. Okay.